Myself, Dr. S. Kandasamy, Associate Professor of Biochemistry, KIMS, Puducherry. Today, we are going to discuss about vitamin B12 metabolism. The vitamin B12, the coenzymes are deoxyadenosyl cobalamin and methylcobalamin. These are the two important coenzymes for vitamin B12. Let us see what are the alternative names for vitamin B12. It is nothing but hematopoietic B complex vitamin, anti-pernicious anemia factor, extrinsic factor of Castell. Here the vitamin B12 as cobalt is linked to choline ring. This is the only unique vitamin because it is synthesized only from microorganisms, not by animal and plants. What are the important aspects of the vitamin B12? The vitamin B12 the only vitamin water soluble that is stored in the body. It is exclusively synthesized by the microorganism. Dietary sources exclusively of animal origin. It is a rare vitamin compound with direct metal carbon bond. Let us see the chemistry of the vitamin B12. The structure of cobalamin is cobalt choline complex, which gives red color to the vitamin. It has 4 pyrrole rings coordinated with the central cobalamin atom to form choline ring. We know that cobalt being a hexavalent atom, 4 nitrogen atoms of the choline rings coordinated with the 4 nitrogen atoms. The fifth valence of the cobalt is attached to the nucleoside side chain, that is dimethyl benzolamazole. The sixth valence of the cobalt is the functional group. Depending upon the functional group, the vitamin name is varies. For example, if it is cyanide group, it is so-called cyanocobalamin. Suppose it's methyl group, methylcobalamin. Suppose hydroxyl group is there, hydroxycobalamin. If it is deoxyadenosyl, it is nothing but deoxyadenosyl cobalamin. Like that. How they are arranged in the vitamin B12. B12 consists of cobalt containing choline ring similar to the ion of the hemoglobin and chlorophyll. Cobalt makes 6 valence. Out of 6 valence of the cobalt, 4 vacancies are occupied by the nitrogen of the pyrrole ring of the choline ring. Fifth valence is attached to the 5,6 dimethyl benzoimidazole moiety. Sixth is free to occupy. The sixth valence is occupied by the so-called cyanide or hydroxyl, adenosyl, methyl group, etc. The group occupying this valence will determine the name of the vitamin B12 coenzyme. Let us see the metabolism of the vitamin B12. If the sixth valence is occupied by the cyanide, it is named as cyanocobalamin, otherwise called vitamin B12A. What is the importance? It is synthetic form, nothing but oral form of the vitamin B12. Suppose 6 valence is occupied by the 5-deoxyadenosyl. The name is 5-deoxyadenosyl, which is a biologically active form. If it is occupied by methyl group, it is so-called methylcobalamin. It is a coenzyme form of the vitamin B12. The 6th valence is occupied by the hydroxyl group. It is nothing but hydroxycobalamin, otherwise called vitamin B12B, which is ingestible form. What are the form of vitamin B12? There are two important active form of vitamin B12, namely 5-deoxyadenosyl cobalamin and methylcobalamin. The 5-deoxyadenosyl cobalamin predominant coenzyme form, which is present only in the mitochondria, which is nothing but storage form of the vitamin B12, especially in the liver and kidney. Whereas another coenzyme form is nothing but methylcobalamin. It is a transport form of vitamin B12 in the serum. It is present intracellularly in the cytosol. What are the dietary sources of vitamin B12? Plant source. No B12 present in the plant. Vitamin B12 synthesis only by the microorganisms. What are the important sources of the vitamin B12? Animal source, liver, eggs, milk, oyster, chicken, food. And then fortified food also contain vitamin B12. What are the? Flour, ready to eat breakfast cereals, bread. Curd is a better source than the milk because lactobacillus in the curd will synthesize vitamin B12. So curd is better source than the milk. Human get small amount of vitamin B12 from the intestinal normal flora. Body stores, especially liver, 4 to 5 milligram of vitamin B12, which is enough for 4 to 6 years. That is a unique property of vitamin B12. What is the daily allowance? Adult male and female need 1 to 2 microgram per day. Children's 0.7 to 1.4 microgram per day. Whereas pregnancy 2.6 microgram per day. The requirement of vitamin B12 during lactation 2.8 microgram per day. Whereas the therapeutic dose is 1 milligram per day orally. These are the recommended daily allowance of the vitamin B12. Now let us see the absorption of vitamin B12 in the system. Vitamin B2 is a unique since it has highly specialized mechanism for absorption. This process is mediated by number of protein throughout the gut. Let us see the detail about the absorption of vitamin B12. Vitamin B12 present the diet as protein bound. This dietary source of vitamin B12 so called distinct factor of gas. In the stomach, pepsin with the help of hydrochloric acid degrade the bound protein and release vitamin B. Then this vitamin B12, which is then binds to salivary vitamin B12 binding protein, so called hepatocorrin or R binder. Hepatocorrin protect the vitamin B12 from acidic medium of the stomach. The R binder and the vitamin B12 complex are transported to the duodenum and cleaved by the pancreatic trypsin enzyme, release vitamin B12, which attach to the intrinsic factor. What happened in the duodenum? Vitamin B12 binds to the intrinsic factor. It is secreted by the parietal cells of the stomach and form the complex called intrinsic factor vitamin B12 complex. This complex travels to the distal ileum and binds to the specific mucosal border receptor called ublin, which leads to absorption of vitamin B12 by endocytosis. This is accelerated by the calcium dependent mechanism. The absorbed B12 is bound to transport proteins called transcobalamin, which then deliver it to the liver and then other parts of the body. In the liver, B12 is converted into deoxyadenosyl B12 and stored about 4 to 5 milligrams stored in the liver, which is sufficient to meet body requirement for 4 to 6 years. Generally, B complex vitamin is not stored in the body. Vitamin is the only vitamin, B12 is the only vitamin and exceptional. Let us see the detail about the different proteins involved, the absorption of vitamin B12 in the system. First, let us see the hepatocorrin, otherwise called R binder. A hepatocorrin or R binder produced by the salivary glands and the parietal cells of the stomach. It is a glycoprotein consists of 433 amino acid and molecular weight is about 48 kilodalton and composed of 30% of carbohydrate. 80% of the B12 is in circulation is bound to hepatocorrin, that is B12 binding protein. What is the function of this hepatocorrin, otherwise called R binder? Function: protect the acid sensitive vitamin B12 while it moves through the stomach. The hepatocorrin also involved enterohepatic circulation. The second one is called intrinsic factor of Castile, otherwise IF. It is a glycoprotein, consists of 8 to 15% of carbohydrate with a molecular weight around about 50,000 dalton. The pH of this intrinsic factor is around 7. The secretion of intrinsic factor is stimulated by food, histamine, and gastrin, whereas it is inhibited by the vagal block. What about the combination between the intrinsic factor and the B12 ratio is 1:1 ratio. Now let's see the transport protein, so-called transcobalamin. There are three types of transcobalamin: transcobalamin 1, transcobalamin 2, and 3. Transcobalamin 2 is physiological active form. It is a beta globulin synthesized mainly by the liver. Half-life is 12 hours. Congenital absence of it causes megaloblastic anemia within a week of birth. And transcobalamin 1 and 3 are also called alpha protein, synthesis by the granulocytes. They act as storage substance for vitamin B2. Half-life is 9 to 12 days. Absence of this transport protein, that is transcobalamin 1 and transcobalamin 3, cause no physiological impairment. Let us see detail about what are the functions of vitamin B12. The first function is it is involved hematopoiesis. That means synthesis of blood, and also involved synthesis of DNA RNA. The third function is synthesis of methionine and choline. Also formation of myelin sheath. And finally, you have the functions of vitamin B12, mainly used for the regeneration of folic acid. What are the active form of vitamin B? We already discussed there are two important active form of vitamin B12, namely deoxyadenosyl cobalamin and methyl cobalamin. Let us the first coenzyme for vitamin B2, that is deoxyadenosyl cobalamin. Deoxyadenosyl cobalamin is a coenzyme for major storage form of cobalamin in the liver mitochondria. The 5' carbon of the 5' deoxyadenosine is coordinated to the cobalt. This is the only known example of carbon metal bond in biomolecule. The second coenzyme form of vitamin B12 is methylcobalamin. Methylcobalamin is a major circulatory form and also found in cytoplasm of the cell. When the adenose group is replaced by methyl group, this is known as methylcobalamin. Let us see briefly about the function of the B12. There are two reactions required vitamin B12 in the system. First one is synthesis of methionine from homocysteine. Here, N-methyl tetrahydrofolate act as a one carbon moiety, donate the methyl group to cobalamin. The cobalamin become methyl cobalamin. This methyl cobalamin donates the methyl group to homocysteine, converted into methionine. This methionine again converted into its active form S-adenosyl. This is a S-adenosyl methionine, otherwise called SAM. This mainly involved transmethylation reaction for various substances like creatinine, neurotransmitter, etc. Suppose due to deficiency of vitamin B12, homocysteine will not converted into methionine. So what will happen? Whatever the methyl group present in the form of N5-methyl tetrahydrofolate stored, this is called that. So the absence of both folic acid and cobalamin leading on to the condition called megaloblastic anemia. The second function of the vitamin B12 is the form of deoxyadenosyl cobalamin. Fats and fatty acid as well as amino acid, branch and amino acid like valine, isoleucine, methionine, threonine converted into propanol. This propanol CoA is converted into D-methyl CoA with the help of propanol CoA carboxylase. Here, biotin as a coenzyme. This D-methyl CoA epimerized into L-methyl CoA. This again converted into succinyl CoA with the alpha, the coenzyme of the vitamin B12 is so-called deoxyadenosyl cobalamin. So succinyl CoA is a end product in this case. This succinyl CoA either enter the TCA cycle or used for synthesis of heme synthesis. That is the second role of the vitamin B12 as deoxyadenosyl cobalamin. Here, due to the deficiency of the coenzyme deoxyadenosyl cobalamin, the methyl malonyl CoA will not convert into succinyl CoA, thereby increase level of L-methanol malonyl CoA in the blood and ultimately excretion of methyl malonyl CoA in the urine. This condition is called methyl malonic aciduria. This is the inborn errors of the due to the deficiency of deoxyadenosyl cobalamin. What are the causes of the vitamin B12 deficiency? First one is nutritional deficiency. The vegetarian diet will cause vitamin B12 deficiency. Another cause is malnutrition. And another cause for vitamin B12 deficiency due to the decreased absorption, like in the case of gastric disorder, achyluria, Vernix anemia, partial or complete gastrectomy, gastric atrophy. Decreased absorption also due to intestinal disorder like ileal resection, Kuru, tuberculosis, pancreatic disease. Drugs which competent for vitamin B12 are metformin, colchicine, which are decrease the absorption of vitamin B12 in the intestine. The fish tapeworm is also cause the intestine cause the vitamin B12 deficiency. So the causes of vitamin B12 deficiency we can discuss under 4, 5 headlines. Nutritional, decreased absorption due to gastric disorder, decreased absorption due to intestinal disorder, drug therapy, and then fish death. What is the how the nutritional deficiency cause vitamin B12? Very common among the vegetarians of low socioeconomic group. The only source for vitamin B12 vegetarian diet is curd or milk. And lower income group may not be able to afford to eat. So let us see something about the Bernix anemia. It is an autoimmune disease. Antibodies are generated against intrinsic factor. So intrinsic factor become deficient, thereby defective absorption of B12 in the intestine. It is characterized by the low hemoglobin level, decreased or no RBC, thereby leading on to neurological manifestation. What are the vitamin B12 deficiency manifestations? We can study under 5 headlines. The first one is folate draft. Second one is megaloblastic anemia. Third one is hyperhomocystinemia. The fourth one is demyelination. And fifth one is subacute combined degeneration. We will see one by one. The first one is folate draft. Vitamin B12 deficiency cause simultaneous folate deficiency due to the folate lack. Therefore, all the manifestations of folate deficiency are seen in vitamin B12. Here, what is folate? Folic acid in the form of N5-methyl tetrahydrofolate donate the methyl group to vitamin B12, that is cobalamin. The cobalamin become methyl cobalamin. The methyl cobalamin again donate the homocysteine. The homocysteine become methionine after obtaining methyl group from methyl cobalamin. Due to the deficiency of vitamin B12, N5-methyl group not getting by the cobalamin. So therefore, homocysteine will not convert into methionine. So folic acid is trapped or accumulated in the form of N5-methyl tetrahydrofolate. This is nothing but folate trap. The second is megaloblastic anemia. The deficiency of folic acid and vitamin B12 leads to the condition called megaloblastic anemia. Megaloblastic anemia differs from normoplastic in two aspects. The first one is size. They are larger. They show delayed nuclear maturation, but normal cytoplasmic hemoglobin synthesis. The causes of the megaloblastic anemia due to the deficiency of both folic acid and vitamin B12 due to impaired synthesis of DL. That is the conversion of deoxyuridine monophosphate to deoxythymidine monophosphate. This leads to arrested cell division in S phase of the cell cycle. This results in accumulation of megaloblasts in bone marrow and release of immune erythrocyte into the circulation. The peripheral smear shows megaloblasts marked variation in signs, that is aniocytosis and shift as well, phycocytosis. And there will be a hyperpigmented neutrophil. The third deficiency manifestation of vitamin B12 deficiency is abnormal homocysteine level. Due to vitamin B12 deficiency, the conversion of homocysteine into methionine step is blocked. Leads to accumulation of homocysteine and cause homocysteinuria. The homocysteine level in blood, high level in blood is related to myocardial infarction. The homocystinemia is one of the independent risk factors for cardiovascular disease. So vitamin B12 and folate are productive against ischemic heart disease. The fourth deficiency manifestation is demyelination. In vitamin B12 deficiency, homocysteine level are high and active methionine, that is methyl donor to various acceptors not formed. Therefore, methylation of phosphatidylethanolamin to phosphatidylcholine is not adequate. This leads to deficiency formation of myelin sheath of the nerve, the demyelination and neurological lesion. What are the biochemical basis of the vitamin B12 deficiencies to neurological system? In B12 deficiency, methyl malonyl CoA cannot convert to succinyl CoA. So there is accumulation of propanol CoA and methyl malonyl CoA. These interfere with myelin sheath formation. The first one, in B12 deficiency, accumulated propanol CoA inhibit acetyl-CoA carboxylase, which impairs fatty acid synthesis in liver and brain, etc., which in turn impairs the myelin formation leading to neurological manifestation. Methylmalonyl-CoA acts as a competitive inhibitor of malonyl-CoA in fatty acid synthesis. Thus, fatty acid biosynthesis required for myelin formation is impaired. B12 deficiency reduces the level of S-adenosylmethionine, which is nothing but active methionine required for transmethylation reaction of the neurotransmitter synthesis. For example, epinephrine, choline. So, vitamin B12 deficiency reduce the level of neurotransmitter. Fifth and last, subacute combined neurological degeneration. This is nothing but Leishman disease. Subacute combined neurological degeneration of posterior as well as lateral column of the spinal cord. Both the sensory and motor tracts are affected. And there is a positive rhombic sign, that means falling when the eyes are closed. And also Rosolimbos sign. This is nothing but percussion of tips of the toe, calves, and exaggerated flexion of the toes. These are the some of the clinical manifestation of vitamin B12 deficiency. Then, what are the clinical manifestations? Both folate and copper deficiency have similar anemia and gastrointestinal symptoms. What are the anemia symptoms? Fatigue, weakness, palpitation, vertigo, tachycardia. Gastrointestinal symptom. So, perforated tongue, weight loss, diarrhea. The oral manifestation of vitamin B12 deficiency initially glycositis having perforated tongue, later gradual atrophy of polypin papilla leads to bald tongue, termed as a hunter or glossitis or Mullis glossitis. And then burning tongues, and finally loss of taste sensation. What are the neurological symptoms? Numbness, paresthesia, ataxia, abnormal reflex, immediate vibrating sensation. So these are the clinical manifestation due to vitamin B12. Let us recollect once again. Both the folate and the cobalamin deficiency have the similar anemia as well as GI symptoms. Anemia symptom, GI symptom, neurological symptom. How will we assess vitamin B12 deficient? We can estimate serum B12 level. Normal B12 level is 150 to 350 picogram per dl. In vitamin B12 deficiency, there will be a decreased serum B12 level. And then peripheral smear, macrocytic hyperchromic anemia, RBCs, aniocytosis, pyglocytosis, hyperpigmented neutrophil. Bone marrow aspiration shows marked erythropoietic hyperplasia. Chilling test shows impaired absorption of vitamin B12. Then Pernicious anemia, reduction of antibody to intrinsic factor, detecting A. calvaria through the gastric juice analysis, absence of intrinsic factor gastric juice. Then increase plasma indirect bilirubin. We can also estimate homocysteine level both in urine and plasma. Then in plasma, the reference range of homocysteine is 5 to 15 micromole per liter. In the case of vitamin B12 level, B12 deficiency, homocysteine level is increased greater than 100 micromole per liter. Then we can also estimate methylmalonic acid both in plasma and urine. The normal level of methylmalonic acid in the plasma is 70 to 350 nanomole per liter. In case of vitamin B12 deficiency, there is increased level of methylmalonic acid in the plasma as well as increased excretion of methylmalonic acid in the urine. And also we can able to estimate transport protein of the vitamin B12, that is hollow PC. These are some of the assessment of the vitamin B2 deficiency the human. What is the difference between folic acid and B12 deficiency? In folic acid deficiency, serum folate level less than 2 nanogram per ml, whereas normal B12, normal methylmalonic acid and elevated homocysteine level. In the case of vitamin B12 deficiency, normal folate level, low B12 level, elevated methylmalonic acid and elevated homocysteine level. Folic acid alone should not given in macrocytic anemia as it may aggravate the neurological manifestation of vitamin B12 deficiency. What are the management part of the vitamin B12 deficiency? Or oral dose in the form of initial doses 1000 to 200 microgram per day for 1 or 2 weeks. Whereas maintenance dose is 1000 microgram per day for life. Whereas intramuscular management under 2000 microgram every day or every other day for 1 to 2 weeks. The maintenance dose under 2000 microgram every 1 to 3 months. Folic acid supplemented reverse hematological abnormality observed in vitamin B12 deficiency, but neurological symptom still persist. So, simultaneous administration of both the vitamin B12, folic acid advised to treat megaloblastic anemia. Thank you so much. These are the reference